Hey everyone, today we are going to start a new chapter, and honestly, one of my favorite subjects in teaching biology, which is population genetics and microevolution. So, what we're going to do in this chapter is we are going to take a lot of the concepts that we talked about in chapter 18 with evolution and natural selection as a whole, and we are going to apply those concepts to very specific situations within individual populations. So, let's go ahead and get into this. So, to kind of introduce you to the idea of what we're talking about here, I always like to talk about methicillin resistant Staphylococcus aureus whenever we're introducing the concept of population genetics. So, MRSA is a strain of common skin bacterium that is very deadly if you contract it. And oftentimes you'll see this being contracted in very sterile environments like hospitals. So, the whole idea is that when bacteria become exposed to antibiotics for very long periods of time, the whole idea of antibiotics is that antibiotics are supposed to kill pathogenic bacteria so that we don't get sick, right? But the idea here, which ties into evolution, is that the presence of an antibiotic provides what we call a selection pressure on the bacteria. So, this is the change in environment that we always talk about that requires adaptation for natural selection. So, if a bacterium gets exposed to an antibiotic, there are two options. Either you are killed by the antibiotic, or you quickly adapt by genetically mutating and you develop a resistance to the antibiotic, which then can be passed on to all of your offspring from asexual reproduction. So those are the two options. Either you survive because you adapted or you die off because you didn't adapt. So that's not really any different than any other type of organism. But in the case of bacteria, this is particularly concerning because in the last 30, 40 years, we have really started to see a over-reliance and overuse of antibiotics. So if you go to the doctor, and if the doctor even suspects that you might have some kind of bacterial infection, just to be safe, you'll probably be prescribed antibiotics, even if they're not absolutely necessary. So, what this does is within the population, it creates this constant selective pressure on any bacteria so that if these bacteria are going to survive, it's in their best interest to start developing these resistances to antibiotics. And if the bacteria develop these resistances, like MRSA does with the antibiotic methicillin and other penicillin derivatives, then you're going to end up with people getting sick with bacterial infections that cannot be treated with any routine courses of antibiotic. And the really concerning thing is that we are finding that the rate of bacteria developing these antibiotic resistances through natural selection, this is really outpacing the rate of new antibiotic production through the pharmaceutical industry. So that's a very concerning figure and something that absolutely needs to be shored up. So shoring that up is going to require a better understanding of evolutionary biology. So understanding how these sorts of antibiotic resistances actually come to be might help us better address these superbug crises. So if you're interested in that topic, there is lots of information out there. I absolutely encourage you to go look into it. Now, in face-to-face lecture, I usually show this video. Since this is a YouTube video in and of itself, I do not believe it would be appropriate for me to show another YouTube video within my own YouTube video. I could probably get into trouble for that, but I absolutely recommend you follow this link here, and I'll try to remember to place this link into the description of this video so that you can follow it on your own. But this is a video that's provided from the Kishoni lab at Harvard University Medical School, showing how quickly bacteria can develop resistances to antibiotics. So this is going to be a very sped up and very rapid version of what we're going to see in this chapter with population genetics. So before you go any further, I would absolutely recommend you take a look at this video. Okay, so let's go ahead and remind ourselves of some terms. A population is a group of organisms of a single species that occupy a particular area at the same time. So this could be all the people in a city, it could be all the dogs in a park, It could be all the birds in a forest. It could be all the bacteria on the surface of your kitchen. So a population refers to one specific species of organism that all occupy the same place at the same time. So if you're specifically talking about sexually reproducing organisms, we've seen before that we ensure genetic diversity through the crossing over and independent assortment processes that are a part of meiosis 1. That's very important, as we saw in the previous chapter, because having a very diverse population ensures that if some selection pressure manifests itself by a change in the environment or something similar, you increase the likelihood that someone within the population is going to have the physical characteristics necessary to adapt and survive. So the gene pool for that population, if genetic diversity is as high as it should be, the gene pool should have a very wide variety of alleles, and all the members of the population should have a wide assortment of genotypes and therefore phenotypes. So what the field of population genetics does is studies the diversity of allele differences and then tracks changes in allele frequencies and phenotypic differences over time. And basically, if we track these things, and then if we do notice that those things do change over time, then that represents sufficient evidence that some kind of evolutionary change has occurred. So when we are speaking specifically about populations of organisms, we prefer to use the term microevolution. So what we want to do here is we want to understand whether microevolution is occurring or not within a population. And as I indicated before, we are going to do that by tracking both allele frequencies and genotype frequencies over time. So a good example of this, and of course you understand that not everything in genetics is going to be Mendelian in nature. Not every gene is only going to have two different alleles. Not every trait is going to be determined by just one gene. But to make this nice and easy to understand, let's consider the case of these black-peppered moths that you're looking at here. So the trait that we are considering here is their body and wing color. So the gene for body and wing color has only two alleles, and these alleles show classical Mendelian complete dominance. The capital D dominant allele codes for black body and wing color, and then the little d recessive allele codes for white and peppered body and wing color. So you can kind of see what those look like right there. So, of course, we are familiar with this by now, especially if you've been following these lectures since we got to Chapter 10. Every diploid organism is going to have two copies of each chromosome. So for each one of these three individual moths here, you can see they each have two chromosomes, one they got from their mother and one they got from their father. So each one of these identical chromosomes has the same genes in all the same places, but because alleles represent gene variants, the alleles may be the same or they may be different on these two homologous chromosome pairs. So you can see that this individual here is homozygous dominant. He has both capital D dominant alleles, so his genotype is capital D, capital D. So he is homozygous dominant and he has black body and wing color. This individual here has one dominant allele and one recessive allele. So this individual is heterozygous, but because of the way dominance is playing out here, this individual has exactly the same phenotype as the moth that we just looked at before. And then finally, this individual here has two copies of the recessive allele. So this individual is homozygous recessive, and because of the way dominance plays out here, since there is no dominant allele, the recessive trait is allowed to manifest itself, and this individual has a white peppered body and wing color. Okay, so before we go any further, we want to introduce some terms associated with microevolution, and we'll explore more of what these things mean a little bit later on. So first, I want to talk about genetic drift. So we said that microevolution, as to whether it's occurring or not, it all boils down to whether allele frequencies and genotype frequencies are changing in a population over time. But what we're going to see is that not every allele frequency and genotype frequency change that we see is necessarily going to be because of natural selection. So microevolution will be occurring if those things change, but it's not necessarily because of natural selection. So if it's not natural selection, the most usual suspect for what's going on is that the population is experiencing what we call genetic drift. So genetic drift represents a change in allele frequencies that is neither advantageous nor disadvantageous for any particular phenotype in the population. So if you're examining a population over the course of, let's say, 100 years, and then at the end of that 100-year period, you notice that the allele frequencies have really changed within the population, that doesn't necessarily mean that one particular allele or one particular phenotype was necessarily good for the environment or bad for the environment at the time. So genetic drift are changes in allele frequencies that are usually the result of some chance event. So not necessarily because one trait was better than the other, but just because it just kind of played out that way because of dumb luck. So the founder effect is a type of genetic drift that, again, we will be exploring a little bit more later on. This is a change in allele frequencies that occurs because an isolated subset of a single population basically migrates out, and then it becomes not reflective of the population as a whole. So in this new population that splits off from the old population, you'll see, you might possibly see alleles that were formerly very rare in the previous population are either going to now be overrepresented in this new population or totally absent. And whether one of those occurs or the other, it just comes down to chance. So we actually saw some of this with the Ecuadorian mainland finches migrating to the Galapagos Islands. The allele frequencies are very different if you compare the mainland finches to the finches that Darwin saw on the Galapagos Islands. So at the time, before any natural selection occurred for the beak sizes, we probably would have seen some allele frequencies right after that migration occurred, just because it's hard to get a perfect representation of what the original allele frequencies were in the original population when you take a small little sample and put it somewhere else. And then finally, we'll eventually talk about the concept of gene flow. When gene flow occurs, you may start to see a change in allele frequencies due to migration of individuals in and out of the population. So this does not itself, like the others, indicate natural selection, but it can help to catalyze the selection process by potentially bringing new alleles into the gene pool or carrying new alleles out of the gene pool. Okay, so now we're going to talk about what is my favorite topic in population genetics, Hardy-Weinberg equilibrium. This is going to involve just a very small amount of math. If you don't like math, don't worry, the math here is not too terribly difficult, but I will walk you through it. So the frequencies of all alleles in the gene pool for any given gene have to add up to 1 or 100%, however you choose to represent that number. So if we are considering one gene that only has 2 possible alleles, like we did in the previous example, then we should have this equation here. P plus Q equals 1. So P would be the frequency of one of the alleles, and Q would be the frequency of the other allele. It doesn't really matter which one is which, right? So microevolution is going to be occurring when these frequencies change significantly over time. So if at the start of time, so if like say 100 years ago, if those frequencies were P is 0.5 and Q is 0.5, and then you fast forward 100 years to today, and now suddenly P is 0.8 and Q is 0.2, then that indicates that microevolution has occurred and something has occurred to change the frequencies of those alleles. But regardless of any microevolutionary change, those frequencies have to add up to 1. The total number of alleles in the population, the frequencies of which have to add up to 1. Now here's an interesting question for you. If P plus Q suddenly add up to less than 1, what do you think that would indicate about the gene pool? So remember that P plus Q equals 1 equation that you're looking at in big bold print at the top of the slide. That applies to a situation in which there are only 2 alleles in the gene pool for that particular gene. So if you fast forward 100 or 1,000 years and now suddenly P plus Q add up to, say, 0.9, that probably tells you something. That's probably telling you that there is now at least a third allele for that particular gene. So some kind of genetic mutation has occurred that has created a new allele within the population. So now instead of P plus Q equals 1, it would have to be P plus Q plus the third allele equals 1, or the fourth allele equals 1. So however many alleles you have for that particular gene, all of their frequencies have to add up to 1. That is what we are trying to say here. So if a mutation occurs such that a new allele is created, then that new allele would have a calculable frequency that has to be accounted for in this calculation. So P plus Q equals 1 makes the assumption there are only 2 alleles in the gene pool for that particular gene, but of course that is not always necessarily going to be true. So if these allele frequencies do not change appreciably over time, then that makes it safe for us to assume that microevolution has not occurred, absent any genetic drift and gene flow effects. Okay, so let's move on to the next part of Hardy-Weinberg equilibrium. So in addition to tracking allele frequencies over time, we also want to make sure we're keeping track of genotype frequencies. So we will see that it is possible for genotype frequencies to change without the actual allele frequencies changing. So it's good to track these things separately. So we do this with what we call the Hardy-Weinberg equation. P squared plus 2pq plus q squared, I forgot to put the plus in there, so plus q squared equals 1. So, like we said, P can equal the frequency of the dominant allele, or it could be the recessive allele. It doesn't really matter which is which. So if P equals the frequency of the dominant allele, then P squared, which is P times P, that would equal the frequency of any homozygous dominant individuals within the population. If Q equals the frequency of the recessive allele, then Q squared, Q times Q, would equal the frequency of homozygous recessive individuals within the population. And then finally, 2pq, 2 times p times q, would equal the frequency of heterozygotes within the population. So if you are keeping track of a gene that only has 2 alleles, there are only 3 possible genotypes: homozygous dominant, homozygous recessive, and heterozygous. So these 3 things added together should give you a frequency of 1, just like P plus Q added up together to give you 1. So this particular equation that you're looking at here really only works for genes that have 2 alleles. So the equation also makes a long list of assumptions that we're going to see are not always necessarily true. So this equation has a lot of limitations associated with it. It can still be very helpful and very useful to us, but we do need to recognize that it does have some serious limitations to it. It only works for genes that have 2 alleles, and we're going to see that there are a laundry list of assumptions that we have to make. Okay, so ignore the bottom part here. I forgot to animate that properly, so just ignore that for now. So let's look at an example here. It says, say you have a population of 25 of those white-peppered moths that we were looking at before. So let's say that 9 of them are black and 16 of them are peppered. So with a total of 25 moths, that means there has to be a total of 50 alleles within the gene pool. So based on the limitations here, we cannot immediately calculate the frequency of the dominant allele, capital D, or the genotype of the black moth, since there are 2 possibilities there. A black moth could either have the genotype capital D, or a genotype of capital D, little d. So we don't know that quite yet, so we're going to have a really hard time going right to calculating allele frequencies. But what we can do is, if we're assuming that this population is stably in Hardy-Weinberg equilibrium, then we can kind of start piecing everything together. The first thing we can do is we can calculate the frequency of the homozygous recessive genotype. Since we know there are 16 peppered moths out of a total of 25 total moths, the frequency of that particular genotype must be 0.64, 16 over 25. Well, since the frequency of the homozygous recessive genotype is equal to q squared, if we take the square root of 0.64, then Q would equal the frequency of the little d allele, which would be 0.8. And then, based on P plus Q equals 1, we could calculate the frequency of the P, which is the frequency of the capital D allele, which has to be 0.2. So then if we piece everything else together, p squared would represent the frequency of the homozygous dominant genotype, which would be 0.04. So that would literally be just one of the 25 moths is homozygous dominant. And then the frequency of the heterozygotes would be 2 times 0.1 times, or 2 times 0.2 times 0.8, which would be 0.32, the frequency of the heterozygotes. So what this tells us is that of the 25 moths, one of them is homozygous dominant, 8 of them are heterozygous, and the remaining 16 of them are homozygous recessive, which of course we already knew. But the nice thing about this is that if you're willing to make a big list of assumptions, which again are not always true, you can get a pretty good estimate of what the genotype distribution is going to be within a population, as long as you have the information that we have up here. How many members are in the population, and how many of them have each particular type of phenotype. So you can see that our numbers actually work out here. So you can see how all these calculations are done in the form of a Punnett square. So if the frequency of the dominant allele is 0.2, then in doing a cross here, you can see that the probability of producing a homozygous dominant offspring is 4%. The probability of producing a heterozygote is 0.16 for this particular possibility plus 0.16 for this possibility here, which would add up to 0.32. So the fact that we have two different possibilities here, that's how we get our 2pq for calculating the frequency of heterozygotes. And then finally 0.8 for little d times 0.8 for little d here gives us the 64% probability of producing a homozygous dominant individual. So if we quickly check our work, we can actually see that we really nailed the population's genotypes and allele frequencies to the number. You can see that of the black moths, one of them is homozygous dominant, 8 of them are heterozygous, and then based on the way the calculations work, we also get the frequencies right as well. 0.2 for the frequency of the dominant allele, 0.8 for the frequency of the recessive allele, 0.64 for the frequency of homozygous recessive, 0.32 for the frequency of heterozygotes, and 0.04 for the frequency of homozygous dominant. But here's the deal. This kind of estimation will only be good if the population is in what we call Hardy-Weinberg equilibrium, which makes, like I said, a big list of assumptions that basically assume that allele frequencies will not change over time. So this population, if it is in Hardy-Weinberg equilibrium, this population can self-sustain itself with sexual reproduction over time, and even so, we still would not expect those allele frequencies to change unless one of those assumptions ends up not being true. So now it's finally time for us to talk about what those assumptions are and why they are usually almost always not going to be true. Okay, so that will be the first thing that we talk about in our next video. So I thank you for your attention as always. Join us next time and we will start diving deeper into Hardy-Weinberg equilibrium and population genetics. See you next time.